Hi, hello friends. In the previous session, we have dealt with all the different types of meristematic tissues based on various classifications. In today's session, we are going to learn about different types of cell theories proposed by various scientists, which describes the growth and development of apical regions in a plant body. Okay? So, before that, we are going to understand about meristematic initials and its derivatives. Meristematic initials are nothing but undifferentiated actively dividing cells which are present in the meristem regions. So, these cells meristematic initials can be in one or two layers conducting both anticlinal and periclinal divisions. Later they will be derivatives, they will give the derivatives of many tissues like epidermis, cortex, vascular bundles and all. Okay? So, yes, if there is only single apical cell present which is undergoing the division activity and giving rise to derivatives, it is called as meristematic cells. If group of cells which are in 1 to 2 layered or many layered are called as apical initial cells, will conduct both anticlinal and periclinal divisions. It will conduct both anticlinal and periclinal divisions and give rise to many different tissues like epidermis, cortex region and stellar region, which includes vascular bundles, pith, pericycle. Yes. Now, if we look into the types of meristematic initials and their derivatives, primarily we have apical meristem, lateral meristem and intercalary meristem. The derivatives from the shoot apical and root apical meristematic initial cells are protodome, procambium, ground meristem. These are the derivatives from shoot apical meristem and root apical meristem. Okay, and the examples include formation of primary tissues like epidermis, vascular tissues and ground tissues, and those cells which are derived from cambial initials like vascular cambium and cork cambium include secondary xylem, secondary phloem and cork. Example, stem thickening and woody stem thickening in woody plants, like it can be gymnosperms or dicotyledons. Next, intercalary meristem, this is mostly used in regeneration, like regenerated lost parts elongate stem and leaves. Okay? Grace leaves regrowing after grazing is the best example for intercalary meristem. You can take a screenshot of this table. Next, based on the position and function of the apical meristem, we have these derivatives like inner part of shoot apical and root apical meristem from procambium initials which are located in inner part of shoot apical and root apical region will give rise to primary xylem and primary phloem. Best example is vascular tissue development. Okay? From ground meristematic initials which are located below the protoderm surrounding procambium. Okay, they are located below the protoderm peripheral region and it is surrounding the procambium. This will give rise to three simple permanent tissues. What are they? Parenchyma, Collenchyma and Sclerenchyma. For example, they are forming cortex, pith and storage tissues. Lastly, it is protoderm initials which are located in the outermost layer of shoot apical and root apical meristems. They give rise to epidermis, which is the protective layer. Formation of stomata, root hairs in roots, trichomes in shoot is formed from protoderm initials only. So, from protoderm initials we are getting epidermis and stomata in leaves, root hairs in roots and trichomes of stem. Ground meristematic initials will give rise to parenchyma, collenchyma, sclerenchyma which are forming out cortex, pith and storage tissues. Procambial initials will give rise to primary xylem and primary phloem which leads to vascular tissue development. This is based on the position and function of apical meristem. Then if we look into based on the lateral meristem activity, okay, based on lateral meristem activity. Fusiform initials which are located in vascular cambium, ray initials also which are located in vascular cambium. They give rise to secondary xylem and secondary phloem, whereas ray initials will help in the tissues, will help in the formation of tissues like vascular rays which conduct radial transport and storage. So, yes, fusiform initials are present in between primary xylem and primary phloem. Here fusiform initials are present. In between two vascular bundles, ray initials are present. Ray initials are present in between two vascular bundles which give rise to vascular ray radial transport and storage. Best examples are nutrient transport in woody plants. Then comes cork cambium initials which is located near phloem, and this gives rise to cork, that is phloem, phloderm which is inner bark. Okay. Then this best example is this gives rise to the formation of bark in trees. So, yes, fusiform initials between primary xylem and primary phloem which gives rise to secondary xylem and secondary phloem. Ray initials which will help in radial transport or conduction, and it is also involved in storage. Cork cambium initials will give rise to cork and phloderm which forms the bark in trees. This classification is based on the lateral meristem activity. Yes. Lastly, if we see the number of initiating cells or apical cells in root and shoot apices. Number of apical cells in both root and shoot apices will divide this into groups like bryophyta, pteridophytes, gymnosperms and angiosperms. Based on number of root and shoot apical cells, they are differentiated. For example, in bryophytes and liverworts, which are primitive plants, only single apical cell is present. This chart is very much important. Okay. So, yes, in primitive plants like bryophytes, mosses, liverworts and horns, wherein single apical cell is present, the growth occurs from one tetrahedral cell. The growth occurs from one tetrahedral cell which is totipotent, and this is pyramidal in shape. This is pyramidal in shape, and this divides to form new tissues. Second, if you see in pteridophytes which includes ferns and lycophytes, single or group of apical cells are present. Single or group of apical cells are present. Here, group of tetrahedral cells will act as initial cells. Group of tetrahedral cells will act as initial cells like this. Okay. These are meristematic initials. Meristematic initials. Then gymnosperms, which includes conifers, cycads, pinus, okay, wherein group of apical tissues, apical initials are in what organization? Tunica-corpus organization. We will now learn it in a more descriptive way. Tunica-corpus type of organization you will see in gymnosperms, wherein meristems are organized to multiple layer, that is tunica and corpus. Angiosperms, that is in flowering plants, multiple initials are present, tunica-corpus organization again. So, in gymnosperms and angiosperms, tunica-corpus organization is seen, wherein shoot meristem is divided into outer tunica epidermis formation and inner corpus body tissue. Now, if we look into various theories that scientists have proposed, which includes apical theory, Histogen theory, Tunica-corpus theory, Mantle-core theory, Anneau initials and Meristema attente theory and Newman concept. These 6 theories we are going to learn with examples. If you look into apical cell theory was proposed by Hofmeister and Nageli. Apical cell theory is proposed by Hofmeister and Nageli, wherein according to this theory, single pyramidal cell in the shoot apex of vascular plants. This cell takes part the shoot formation and its development. Single pyramidal cell will take part the shoot formation and its development. Apical cell theory is found appropriate in case of some algae, bryophytes and pteridophytes. The main drawbacks of this apical cell theory is this theory is not applicable to higher plants like higher classes of plants like gymnosperms and angiosperms due to the presence of complex meristematic tissue. In them, how the meristematic tissues is arranged? The apical initial cells are arranged like in Tunica-corpus. Tunica, multiple layers, Tunica-corpus, they are arranged, okay, in gymnosperms and angiosperms. So, apical cell theory is not applicable to gymnosperms and angiosperms. Next, if we consider Histogen theory is the second theory we have to know. The scientist who proposed Histogen theory is Hansteen. Scientist who proposed Histogen theory is Hansteen. He said that group of initial cells, there are many layers of initial cells. In each layer there are group of initial cells, you will name it as histogen. Group of initials, you will call it as histogen. As such, 3 layers are there, which is dermatogen, which is also called as calyptrogen. Dermatogen, which is here, which is also called as calyptrogen. Next it is peribulum. Next layer is peribulum and inner layer is pleurum. As such, we have 3 histogens. 3 histogens, dermatogen or calyptrogen, second is peribulum and third is pleurum. So, now if we look into dermatogen, which is the outermost single layer and it will form epidermis of the shoot. Peribulum, it is the middle layer and cells which are present in the peribulum are isodiametric cells. Cells which are present here are isodiametric cells, and pleurum, it is the innermost layer which gives rise to vascular cylinder, and in turn it comprises of primary xylem, phloem and medullary rays. So, if we look into 3 cells, 3 layers of histogens, dermatogen will form epidermis. What type of divisions it will conduct? Only perpendicular divisions, those are anticlinal divisions, and this will leads to the surface expansion. Second is peribulum. Second is peribulum, which forms the cortex. Peribulum forms cortex. It will undergo both anticlinal and periclinal divisions, which leads to growth and differentiation of cortical cells. Thirdly, it is pleurum. Third is pleurum, wherein vascular tissues, it forms, pleurum forms vascular tissues and central region. It forms vascular tissues and central region. Mainly it undergoes periclinal or parallel divisions, and the function it conducts is longitudinal growth and vascular tissue formation. It conducts the function which is longitudinal growth and vascular tissue formation. The drawbacks, major drawbacks of this is there is no clear distinction between dermatogen and peribulum. There is no clear distinction in dermatogen and peribulum, which are 2 inner layers. These tissues, dermatogen and peribulum, are supposed to be produced by particular histogen, were sometimes formed by another histogen, which means nothing but the dermatogen which is supposed to produce epidermis, dermatogen which is supposed to produce epidermis will also produce some layers of the cortex. This is producing cortex also. So, there is no clear differentiation between these 2 layers. That is one major drawback. No proper distinction between dermatogen and. So, next is Tunica-corpus theory, which is very important. This is present in more distinct form in higher plants like gymnosperms and angiosperms. So, this theory is given by Schmidt in the year 1924. This theory is given by Schmidt in the year 1924. So, here we have, according to this theory, we have 2 zones in the shoot apex. Shoot apex has 2 zones, that is the outer layer which is said to be tunica and inner mass is corpus. Outer is tunica and inner mass is corpus. Tunica will conduct only anticlinal divisions. Tunica will conduct only anticlinal divisions, and this will lead to increase in the surface expansion of the plants and increase in epidermis, whereas corpus conducts both anticlinal and periclinal divisions, which will increase the thickness and volume of the plant. So, tunica cells conducts only anticlinal divisions and it will increase the surface expansion but not thickness. Okay. Next, tunica conducting anticlinal divisions will form two tissues, that is epidermis and outer cortex layers. Second is corpus layer. Corpus will develop from only one tier of initial cells. Corpus develops from only one tier of initial cells, which divides in all the planes in a sense, both anticlinal and periclinal divisions. We know what are anticlinal divisions? Perpendicular. Periclinal are parallel to the surface. So, yes, the tissues formed because of these two divisions in the corpus will lead to the formation of inner cortical layers, vascular tissues and pith. These three are formed as a result of division in corpus. Now, based on the divisions and what tissues are formed over here, it is there, and on the basis of this theory, many types of tunica corpus are identified in vascular plants. First type is plants with no difference in tunica corpus shoot apex. There is no difference between tunica and corpus shoot apex. For example, there is no clear distinction is seen in pteridophytes. There is no clear distinction seen in the primitive vascular plants like pteridophytes, wherein all the cells divide into multiple planes without forming tunica and corpus. In pteridophytes it is seen, wherein it has only monoplex, means only single layered, which is dividing into all the planes. There is no proper differentiation of two layers, tunica and corpus. Second, if you see in some gymnosperms, plants like cycas, they do not show any difference between tunica and corpus. Okay, in few gymnosperms like cycas, there is no differentiation seen between tunica and corpus. Second is plants with partial distinction between tunica and corpus. Okay, only partial distinction is observed, wherein gymnosperms generally they have mostly homogeneous shoot meristem, wherein cells are not much differentiated. Some primitive gymnosperms like cycas and gingo, primitive gymnosperms like what? Cycas and gingo will show no tunica corpus distinction. They do not show any tunica corpus distinction, while in needles and some conifers will exhibit partial differentiation. Okay. Primitive gymnosperms like cycas and gingo, they do not show any tunica corpus distinction, while needles and some conifers will exhibit partial differentiation. Coming to needles, Neetum, Ephedra, Wilwichia. Among gymnosperms, needles show most angiospermic features, including some distinction between tunica and corpus. Primary distinction is shown in needles. Outer layers will divide anticlinally, while inner layer will show mix of divisions which include both anticlinal and periclinal divisions. Based on their behavior of division, primitive partial differentiation is seen between tunica and corpus. Next, if we observe Araucaria, this is one example which is a type of conifer in which, just a second, Araucaria, wherein the superficial cells include tunica like zone and inner we have corpus like mass. Superficial cells include tunica like zone and inner corpus like mass we can observe. Next is taxus. Taxus is an example where you can see two layered shoot apex. In taxus you can see two layered shoot apex, wherein outer layers behave like tunica and inner layer will show mixed divisions. Lastly, some Pinaceae members like pines and firs wherein some species of pines and abies, the shoot meristem shows a slight stratification wherein outer will divide anticlinally, and there is this distinction is not as strong as in angiosperms, but outer is dividing anticlinally, inner is showing anticlinal and periclinal divisions, but there is no clear differentiation as how it is in angiosperms. So, partial differentiation is seen in knee tails, Araucaria, Taxus and Pinaceae members like Pinus and Firs. These examples we need to remember, that is it. Third is Tunica-corpus layers, they will show distinct difference in dicotyledons. For example, Arabidopsis thaliana. Wherein, just a second, Arabidopsis thaliana, wherein the outer tunica is 1 to 2 layered, and inner corpus forms the inner mass of the cells. Tunica strictly it is conducting anticlinal divisions for surface expansion. Corpus, of course, it is conducting two types of divisions and helping in volume growth. Second type is sunflower, which is Helianthus annus. Here also you can see 2 layers of tunica and 1 layer of corpus, wherein layer 1 and layer 2 will act for tunica, and third layer, layer 3 is for corpus. Clear? Next, so this corpus divisions will increase vascular tissue and pith. Next type, if we observe tomato. Tomato is an example again, that is Solanum lycopersicum. You can see clear divisions, but in here what we need to remember is tunica is 3 layered. Tunica and corpus is 3 layered, wherein only third layer will show mixed type of divisions, forms vascular tissue and central pith. Second layer also is showing 2 type of divisions, which shows or which leads to the formation of ground tissue. Okay. So, yes, only layer 1 is conducting anticlinal divisions, 2 and 3 both anticlinal, periclinal, forming ground tissue, vascular tissue and central pith. Then comes to monocotyledon plants. Best example is maize, that is Zea mays. Same here, layer 1 forms epidermis, 2 will form mesophyll cells and peripheral vascular tissues, 3 forms vascular tissues, pith and medullary rays. What layer is forming which type of tissues by conducting what type of divisions, be it anticlinal or periclinal, this is most important. Next, rice. In rice, if you see, similar to maize, there are clear differentiation. The corpus layer in rice, the corpus layer will prefer a major role in forming stem elongation and leaf primordia. Okay. Then drawbacks of Tunica-corpus theory include Popham and Chan did not accept Tunica-corpus theory as they did not recognize the relationship between the origin of these 2 tissues. Okay. The origin of 2 tissues, that is tunica and corpus, are not distinct, that is why these scientists did not accept that theory. But the significance of Tunica-corpus theory includes, it provides a structural and functional model for how different layers of meristems contribute for organ development, and then it is useful in understanding specific growth patterns. Okay? Yeah. Then fourth theory is mantle-core theory. Here the mantle layer is outer layer and core is the inner layer. Here mantle is observed to conduct only anticlinal divisions in the mantle region of monocots. Okay. This is proposed keeping monocots in view. They have proposed this theory, and it is proposed by Popham and Chan in the year 1950. Outer layer of cells in swollen region of shoot apex are known as mantle. Outer layer of cells are known as mantle. Here in mantle region, anticlinal divisions are observed. Inner core, they observed anticlinal divisions in mantle region of monocots, and inner layer of cells is called as core. It is the scientist B. Bouvet in the year 1952 and 1955. He used this concept to describe the structure and function of shoot apex in angiosperms and some gymnosperms. In some angiosperms and gymnosperms, he has discovered this theory. Next, mantle, which is the outer region, which is the superficial layer. It consists of small dense cytoplasmic cells. They divide anticlinally, which is perpendicular to the surface, corresponds to the tunica of mantle. In the mantle-core theory, it is corresponding to tunica region of Tunica-corpus theory. This gives rise to epidermal and subepidermal layers. Then comes the core. Second layer is core, which is inner mass, which is located beneath the mantle, consisting of larger vacuolated cells that divide in various planes, be it anticlinal and periclinal. So, this corresponds to corpus layer of Tunica-corpus theory. The major role, it plays major role in stem elongation and formation of vascular tissues, cortex and pith. Here are the major distinctive features between mantle and core. You can take a screenshot of this. This just now we have discussed. Next, significance of mantle-core concept. This theory explains the clear differentiation, explains the clear activity of meristem and its growth. Second, it helps in understanding the tissue differentiation, be it epidermal layer formation or cortical layer differentiations or vascular tissue differentiation. Third, it overcomes the limitations of Tunica-corpus theory because it is using both cytological and functional characteristics. Then it explains the variability among different plant species. For example, some plants, especially monocots and certain gymnosperms, have variable shoot apex structure where the Tunica-corpus model is not distinct. But in mantle-core concept, this helps to explain their shoot apex organization. Okay? So, mantle-core concept is explaining the pure shoot apex organization. Next, here in this table you can see clear differentiation between mantle-core theory and Tunica-corpus theory. Okay, you can take a screenshot of this, just now we have discussed about this. Next comes the theory of Anneau initials and Meristem D attente theory, wherein this theory, which is proposed by French botanist, that is Bouvet. Okay, this theory is proposed by French botanist, that is Bouvet, in the year 1952, wherein on the basis, this theory is on the basis of theory of phyllotaxy, which is called as plastocron theory, okay, which is proposed by Pantifol. Okay, on the basis of this theory, Anneau initials Meristem D annate theory is proposed. This theory is supported by contemporary scientists also. The Anneau initials and Meristem D theory explains organization and function of shoot apical meristem in certain plants, particularly in relation to growth regulation, organ formation, cell division patterns. All these areas it will cover. Yeah, Anneau initials are nothing but the ring initials. Anneau initials are nothing but ring initials which are found in the annular zone of initial cells, which are found in the shoot apex. These Anneau initials are found in the shoot apex. These initials are responsible for continuous production of new cells. They are responsible for continuous production of new cells for organogenesis. They surround certain inactive region of meristems, which are called as Meristem D attente. Okay, this is called as waiting meristem. Okay, they are surrounded by Meristem D attente cells. Okay, these are called as waiting meristems, and annual initials are called as ring meristems. Ring meristems are surrounding this meristem initials. Okay, which is called as waiting initials. To be precise, ring initials are covering this waiting meristems. Okay, ring initials are covering the waiting meristems. Functions of Anneau initials include they actively divide to form new lateral organs. They actively divide to form new lateral organs like leaves, buds and flowers. They ensure the continuous shoot growth and development. Then they work in coordination with waiting meristems, that is Meristema D initials, to regulate the growth. Okay. Then Meristem D initials, which is also called as waiting meristem theory, which refers to central inactive zone, which is nothing but, it is nothing like these cells will not divide, no, but the division speed of annual initials is more than the waiting meristems. Okay. Yeah, waiting meristematic zone waits the coverage of vegetative phase into reproductive phase, then it will start on dividing. It acts like reserve of undifferentiated cells, means meristematic cells ready to be activated with rapid growth when needed, okay, when needed during flower formation. Major functions of Meristem D attente or waiting meristems is it maintains the balance between active and dormant growth. Second, it ensures controlled and synchronized organ development. Third, it prevents excessive or uncontrolled division, and which includes the lead to developmental abnormalities. It will prevent the developmental abnormalities by having proper balance between active and dormant growth. Yes. The significance of this theory is it explains shoot apex both continuous and regulated growth. It helps in understanding how plants will switch between vegetative and reproductive phase of growth. Third, it provides insight to plant growth regulation and adaptation to the environmental conditions as well. The sixth theory we are going to learn is Newman concept, wherein it is proposed by Newman in the year 1965. According to this theory, there is no initial cell. There is no initial cells in the apical regions. Instead, the meristematic cells are formed in sequence at various zones. Okay. He termed it as continuing meristematic residue. He said it as continuing meristematic residue. Yeah. Newman divided shoot apex into 3 zones. Those are central mother cell zone, second is peripheral zone, third is rib zone. Okay, central mother cell zone, peripheral zone and rib zone. This is located at the center of shoot apex. If this is the shoot apex, this is located at the center of shoot apex. Rib zone is located beneath the central zone. Peripheral zones are towards the side. Okay. So, this is located at the center of shoot apex, composed of large and vacuolated cells with low mitotic activity. It is composed of large and vacuolated cells with low mitotic activity. Then it functions as reservoir of meristematic cells. It functions as reservoir of meristematic cells that supply cells to other zones as well. Comparable to Meristem D annate, that is waiting meristem in this zone, central zone is compared with waiting meristem of Anneau initials theory. Okay. Then peripheral zone, as we have discussed, which is at the sides, okay, forming a ring around the central zone. Okay, it forms a ring around the central zone, contains actively dividing cells that initiate leaf and auxiliary bud primordia. Third, it shows high mitotic activity, supports continuous organ formation and functionally similar to Anneau initials. Those are ring initials. Third type of zone is rib meristematic zone, which is located beneath the central mother cell zone, characterized by cells dividing in longitudinal rows. This is characterized by cells dividing in regular longitudinal zones, giving rise to pith and central stem tissues, giving rise to pith and central meristem tissues. This ensures stem elongation, and vascular tissue formation. This ensures stem elongation and vascular tissue formation. Then types of shoot apical meristem based on according to Newman. Okay, according to Newman we have 3 types. One is monoplex structure, second is simplex. In monoplex there is symbol one cell which is dividing in all the planes. Simplex, first layer is formed as a result of anticlinal division, second layer is because of periclinal divisions. Duplex wherein first layer is formed because of anticlinal and rest 2 layers of periclinal divisions. Clear? Now we will understand it more better. Monoplex type wherein entire shoot apex is undifferentiated, where there is no clear histogenic cell layer of separation only. Cell division occurs randomly throughout the meristem and occurrence found in pteridophytes and lower gymnosperms like Lycopodium and Cycas. Significance, it represents the primitive type of shoot apex seen in early land plants. Next is simplex type, which shows initial distinction between outer and inner layers, wherein outer layer will show anticlinal divisions, inner layer will show both anticlinal and periclinal divisions. Inner layer will show both anticlinal and periclinal divisions to increase the thickness or volume of the plant body. Third, it occurs, where does simplex type occur? In the plants like gymnosperms, which includes Pinus and Ginkgo. The significance is it acts as transitional form between monoplex and duplex, okay, which gives us an evolutionary significance. Third is duplex type, wherein clearly differentiated 2 or more layers are seen forming Tunica-corpus system, wherein tunica divides anticlinally, corpus both anticlinal and periclinal divisions. Best examples in angiosperms like siamase, dicot plants like sunflower. Then the major significance is this shows the most advanced shoot apex which support the efficient organogenesis in the plants. This is the clear differentiation between monoplex, simplex and duplex type of theories. You can take a screenshot of it. Examples and occurrence, type of division is more important. Then histological organization of shoot apex. From here we will deal in the next class. In next class we are going to deal the histological organization of shoot apex and most importantly signaling pathways. So don't miss it at all. Okay. Thank you.